You have already studied the behavior of chromosomes during cell division. You have also studied that chromosomes are hereditary vehicles on which genes are located. A chromosome is a DNA molecule with associated proteins. Now we will study the structure and functions of human chromosomes. Chromosomes, the carriers of heredity. The term chromosome means colored body. Greek, chrom means color and soma means body. Chromosome can be defined as a strand of DNA molecule with associated proteins. Chromosomes are hereditary vehicles found in the nucleus of a cell. The chromosomes contain genes, thus they are carriers of heredity. The characteristics travel from parents to the offsprings in the form of genes situated on the chromosomes. The genes are composed of DNA and proteins. DNA, or deoxyribose nucleic acid, functions as the genetic material. It forms the chemical basis of heredity. A gene, or segment of DNA, codes for the synthesis of a specific protein which controls the expression of a particular characteristic in an individual. Thus, the gene is the basic unit of heredity found on a chromosome. Chromosome number. The number of chromosomes is constant for all individuals in a species, and each one of them has a fixed and equal number of chromosomes. The number of chromosomes in the somatic cells of higher plants and animals is known as diploid number, represented by 2n. In gametes, sperm, and egg, it is known as haploid number and is represented by n. The chromosome number of some plants and animals is given in the table. Structure. Chromosomes differ greatly in their size and shape. A person may have different sizes of chromosomes. The chromosomes may differ in the position of the centromere. Centromere is the point on the chromosome where spindle fibers are attached during cell division. If the centromere is near the middle, the chromosome is metacentric. If the centromere is towards one end, away from the center, the chromosome is acrocentric. If the centromere is very near the end, the chromosome is telocentric. In addition to a centromere, there may also be a secondary constriction on a particular chromosome. The part of the chromosome located distal to, or far from, a secondary constriction is called a chromosome satellite. As already mentioned in the previous section, each chromosome at the start of cell division consists of two chromatids joined at some point along the length. At the point of joining, a constriction is formed, called centromere. The spindle fibers are also attached to the centromere at the time of cell division. These spindle fibers contract and help in separation of two sister chromatids towards the opposite poles in a dividing cell. On completion of cell division, mitosis followed by meiosis, the chromatids are called chromosomes. These chromatids decondense and form the very thin thread-like chromatin fibers. Each chromatin fiber is made up of DNA and histones with small amount of RNA. Let us now focus on chromatin. Chromatin. The chromatin material consists largely of two strands of deoxyribonucleic acid, DNA, and proteins. 1. DNA or deoxyribonucleic acid. 2. Histones, protein. DNA forms about 40%, while histones form about 60% of the overall part of the chromosome. DNA has negative charges along its length, while histones are positively charged basic protein molecules which are bound to it. This DNA-histone complex is called chromatin. It is shown that the DNA helix combines with group of 8 histone molecules to form a structure known as nucleosome. It has an appearance of beads on a string. In a human cell, there are about 2 million nucleosomes among 46 chromosomes. A molecule. Rosalind Franklin, for the first time in 1953, studied the shape of a DNA molecule. Then, Watson and Crick in 1953 worked out the structure of DNA. Watson and Crick showed that DNA is a large molecule or macromolecule consisting of 2 polynucleotide strands, complementary in nature, wound around each other in a double helix. The strands run in opposite directions, that is, they are antiparallel. Each single DNA strand is composed of repeating nucleotides. Nucleotides are made of 3 components: a phosphate, a sugar, pentose, arranged lengthwise, and a nitrogenous base attached to the sugar inwards. The sugar-phosphate backbone has nitrogenous bases arranged at right angles, giving a ladder-like arrangement. There are 2 purine bases and 2 pyrimidine bases. The bases are guanine, G, thymine, T, adenine, A, and cytosine, C. The guanine is complementary to cytosine and thymine to adenine. How are new DNA strands formed? Formation of a new DNA molecule is called DNA replication. During replication, DNA double helix opens at one end, freeing the strands. To each of these few strands, new complementary strands begin to form in the opposite direction. This process takes place for the whole length of DNA. And autosomes. In human beings, Out of 23 pairs of chromosomes, a specific pair, that is, the 23rd pair of chromosomes, determines the sex of the individual. These are called sex chromosomes. All other 22 pairs of chromosomes are termed as autosomal chromosomes or autosomes. These autosomes carry genes which control somatic traits and have no bearing on the sex. The two members of each pair of homologous autosomes are similar in size and shape. But this may not be true for the sex chromosomes. In human males, one sex chromosome is smaller than the other. The larger one, known as the X chromosome, and the smaller one, known as the Y chromosome. Thus, the condition in the male may be briefly expressed as XY and in female as XX. The sex chromosomes of human females are described as homomorphic, and that of human males are heteromorphic. The human females produce only one type of gametes, all with X, and are said to be homogametic. The human males produce two types of gametes, X type and Y type, and are described as heterogametic. Functions of chromosomes. Chromosomes are hereditary vehicles that contain genes. All the hereditary information is located on the genes. Chromosomes control the synthesis of structural proteins and thus help in cell division, cell repair, and cell growth. By directing the synthesis of enzymatic proteins, chromosomes control cell metabolism. Chromosomes guide development and control cell differentiation. Sex chromosomes XX and XY or XX and XO chromosomes determine the sex of individuals.